Hi, and welcome to my presentation on Chapter 7, Cell Structure and Function. So there's two major types of cells in the world, which are prokaryotic cells and eukaryotic cells. Literally, prokaryote means before nucleus, and eukaryote means true nucleus. So eukaryotes are all of the cells that have a nucleus, and prokaryotes are cells that do not have a nucleus and actually evolved before eukaryotes. There's two major types of prokaryotes, which are bacteria and archaea. So when you divide all of life into kind of groupings based on how closely related organisms are, there's three major groupings that emerge, which we call domains. And two of those domains are bacteria and archaea. Then the third domain is eukarya, and that's the eukaryotes. In addition to not having a nucleus, prokaryotes also don't have any other membrane-bound organelles in their cells. An organelle is basically sort of like an organ, but for a cell. So it's a little sac within a cell. If it's membrane-bound, then it's a little sac within the cell that has all of the components that are needed to do some particular function for that cell. Eukaryotes have a nucleus, which is an organelle, and they also have a lot of other membrane-bound organelles. So there's a lot of membranes inside the cell of a eukaryote. For a prokaryote, in general, there's not any membranes inside the cell. You just have the cell membrane itself, and then the contents of the cell are kind of all swirled together in a way. There are a few exceptions to that. So there are some prokaryotes that have membranes inside the cell. And some of those kind of membrane systems are actually considered to be organelles, but in general, prokaryotes do not have membrane-bound organelles. Then kind of the final difference between the prokaryotes and the eukaryotes is just that prokaryotes are really small cells and eukaryotes are bigger cells. Then I guess I'll mention that for the eukaryotes, like in domain eukarya, there's a number of different types of eukaryotes. Which includes the animals, the plants, fungi, and also single-celled eukaryotes, which would be algae and protists. So just a little more detail on organelles. Like I said, it's like a little organ inside of a cell. So it's a compartment inside the cell where you have everything you need to carry out some specific task. Most organelles are surrounded by a membrane, and we call those membrane-bound organelles. But there are some organelles that do not have a membrane. Usually you find organelles in eukaryotes. There's just a few exceptions to that. And of course, the defining organelle in a eukaryote is the nucleus itself. Organelles bring benefits to the cells that have them. Basically, it lets you get a really high concentration of all of the components you need for some task, within a small area if you put all those components within an organelle. When the concentration of those components goes up like that, then the reaction speed can go up because you're basically increasing the substrate concentration and or the enzyme concentration just by storing everything in one small spot. Also, sometimes cells need to get two things done that kind of don't go with each other. So for instance, cells need to make proteins, but they also need to be able to kind of break down and recycle old proteins or damaged proteins. So those two tasks, making proteins and breaking proteins, they don't really go together. They're going to use different enzymes, and you wouldn't want to have the enzymes that break down proteins just kind of floating around in the cell because they're going to break down all your proteins. So by having organelles, you can just kind of separate tasks that don't mesh well together from each other. And for instance, the cell can keep all of its enzymes that break stuff down sequestered in an organelle so that they don't damage the rest of the cell. Also, eukaryotic cells are just, they're really large compared to prokaryotic cells. So they have actually a lot of volume compared to their surface area, and it can actually be difficult for them to take in enough nutrients to fill up the entire volume of the cell. So by having organelles, they actually don't need to fill up like the whole volume of the cell, with any given nutrient. They might just need to fill up, a particular organelle, which is going to have a much smaller volume. So that makes it easier for them to do that. And the last point I'll make about organelles is just that in general, the space inside of an organelle that's enclosed by that membrane is called the lumen. For some organelles, you also have like a different name for the space inside of it, like a more specialized name that's just for that organelle. But in general, the space inside an organelle is the lumen. Here I have just kind of a diagram comparing a typical prokaryotic cell on the left to a typical eukaryotic cell on the right. So in the eukaryotic cell, you can see the nucleus is this kind of purple ball here, well, ball enclosed by purple in the middle of the cell, and then you have layers of blue membrane wrapping around it. So that's all an organelle that's wrapped around the nucleus. Then you have another organelle here in kind of orange, some more organelles in kind of purplish or pinkish colors. So this cell is basically filled up with organelles and, membrane systems that are involved with those organelles. If you look at the prokaryotic cell, you don't see any membranes inside of it. Instead, it's just kind of globs of stuff. So all this purplish stuff is actually the DNA of the cell, and then here the cell has a little ball of fat in it that it's storing, but it's not enclosed in a membrane at all. So anything that's in the cell is just kind of floating around in here. This diagram is not really showing the scale of the cells correctly. It makes it look like the prokaryote is about the same size as the eukaryote, but actually it would be much, much smaller. So if you look at the scale bar, for the prokaryote, this distance is 0.5 micrometer, and for the eukaryote, the same distance is 10 micrometers. So the eukaryote is about 20 times as big as the prokaryote, actually. So now we'll go through prokaryotic cells in more detail, and then eukaryotic cells, we'll do those in more detail too. So actually these next slides are specifically focusing on bacteria because they're a lot better studied than archaea are. Archaea are pretty similar to bacteria, but they also have some differences from bacteria. In a way, they have some structures that kind of make them look a little bit more like a eukaryote. So they're kind of maybe like a middle ground between bacteria and eukaryotes. But they're just not as much as known about them. So we'll be focusing on bacteria. So inside a bacterial cell, you have several characteristic structures. First, you have the cytoplasm, which is just everything that's inside the cell. Eukaryotes also have a cytoplasm, but in eukaryotes, it's not literally everything inside the cell, it's everything except for the nucleus. But since prokaryotes don't have a nucleus, the cytoplasm is just everything that's contained within the cell membrane. Then you also have the cytosol, which is similar to the cytoplasm, but a little bit different. The cytosol is actually the liquid part of the cell, and it's actually a really thick liquid, so it's kind of more like a jelly. So that's basically the water that's in the cell and also everything that is dissolved in that water. So you're going to have a lot of ions in there, small nutrients, vitamins, enzymes, other proteins, just anything that can dissolve within the water of the cell is going to be a part of the cytosol. Then you have the nucleoid. The nucleoid is the part of the cell or the part of the cytosol that has the DNA of the cell. In prokaryotes, the DNA is always contained on just one big chromosome and is shaped like a circle. So they basically have just one large circle of DNA in them that's containing almost all of their DNA. And that circle is so big that they do have to pack it to make it fit. So it's actually going to be coiled around itself over and over again until it ends up supercoiling around itself. But it's not packed around histone proteins the way that DNA is for a eukaryote. So the way it's packaged is like a little bit simpler. So most of the DNA in the cell is contained within the nucleoid, but there's also in some bacterial cells a little bit of DNA that's not part of the nucleoid, which would mean it's not part of that one big chromosome. Instead, it's contained on a plasmid. A plasmid is a little circle of DNA that's separate from the main chromosome of the cell. Some bacteria don't have any plasmids, some have a lot of plasmids, so it just kind of depends on the specific individual bacterium. And actually some bacteria are able to share plasmids with each other, so they can actually exchange genetic information that way. Then finally you have ribosomes, which are of course the protein complex that also includes rRNA that is involved in actually making protein from mRNA. So in this diagram here of a typical bacterial cell, everything that's inside of the cell membrane is the cytoplasm of the cell. Then this yellow stuff is the cytosol. In blue, you have the nucleoid. So this chromosome here in the nucleoid is packaged up and it is just kind of floating in the cell, but it'll actually kind of stick to this one area of the cell. It is like semi-organized, so it won't just float freely within the cell, but it'll be kind of stuck in one part of it. Then you also have plasmids. This little circle here is a plasmid of DNA, and then all of these little red dots are the ribosomes of the cell. On the top here you have an E. coli cell that has been treated so that its DNA kind of spills out from the cell and becomes unpackaged, and it's this little yellow line here. So all of this line is a single molecule of DNA. This is the chromosome of E. coli. That's kind of how big it is when it's not all packed up. So as you can imagine, the cell has to pack this really tight to make it fit inside. Then the last two structures that you have inside bacterial cells are the cytoskeleton and inclusions. The cytoskeleton is a framework of proteins that is basically just there to give the cell shape and help organize it a little bit. So for instance, it's actually the cytoskeleton that's making sure the nucleoid kind of stays in its proper part of the cell. What the cytoskeleton looks like is going to depend on the specific type of bacterial cell, and there's a lot of different shapes that bacterial cells can have actually. So it's not actually shown on this diagram, but it is shown up here. So here we have 3 different bacterial cells that all have different shapes, and their cytoskeleton is shown. So on this ball cell, the cytoskeleton is actually just a ring around the cell. And in this rod, you have that ring at the center of the cell, but you also have another kind of spiral of cytoskeleton looping down the cell. And then in this kind of curved cylindrical bacterial cell, you have rings that are kind of like spaced along the length of the cell, and you have also a rod or a bar maybe that's traveling the length of the cell. So those are all parts of the cytoskeleton for those cells that are helping them maintain their shape and organization. Then lastly, you have inclusions, which is just any ball of stuff that is stored in the cell that is not dissolved in the water of the cell. So it's not part of the cytosol of the cell because it's not solubilized. So that can include like fat or starch or some other carb that the cell is storing. There's actually a lot of different types of things that different bacteria will store in an inclusion. In this diagram here, the inclusions are shown as these kind of little balls in here. Then in some bacterial cells you might see other structures inside of them. That's not as common, but there are some bacteria that actually have membranes inside of them that would be specialized for carrying out some specific task. For instance, this cell here is a bacterial cell that actually does photosynthesis, and you can see these lines across the cell. These are actually membranes. So this cell has membranes going all the way across it that are just kind of stacked throughout the cell. And in each of those membranes, it has special enzymes that it needs to do photosynthesis. So actually a number of bacteria that do photosynthesis will have membranes inside the cell that have the enzymes that they need for that. And they can be in kind of different shapes, different structures. And then some bacteria have membranes that they use for other things as well. For instance, sometimes even for storing stuff. So those are the internal structures that you would typically find in a prokaryote, in a bacteria specifically. You also have external structures in bacteria that are different from external structures in eukaryotes. First, you have the glycocalyx. which is basically like a layer of jelly or goo that's surrounding the cell outside of its cell membrane and cell wall. And we'll actually talk about the cell wall in a moment, but the cell is first enclosed by the cell membrane or the plasma membrane. Then outside of that, you're going to have a cell wall. Then outside of that, you may have a glycocalyx. Not every bacteria has a glycocalyx. It just depends on the species. There's kind of two types of them. Some of them are more solid, some of them are more kind of slimy. In either case, it's made out of sugars and proteins, and the role of it is just to protect the cell. So it kind of makes it harder for the cell to dry out, and it makes it harder for toxins to get into the cell. In addition to the glycocalyx, you also have flagella that some bacteria have, which is just basically a long whip that the cell can rotate like it's a rotor, and it lets them move through liquid really quickly. Then you also have fimbriae and pylae, which are kind of similar to each other. Fimbriae are just little sticks coming out of the cell that are sticky, and they let the cell grab onto stuff. Pylae are similar, but they're longer. They're actually kind of like a special type of fimbriae that are longer than regular fimbriae. And they have different roles, but one of the most important ones would be to actually allow bacteria to transfer DNA between each other. So a pilus is actually hollow in the middle, and if one bacteria kind of sticks another bacteria with its pilus, then it can actually send any plasmids that it has down that pilus into the other bacterial cell. So that's one way for bacteria to exchange genes with each other. So flagella, fimbriae, and pylae are all plural words. Flagellum is the singular for flagella. Fimbrium is the singular for fimbriae, and pilus is the singular for pylae. With all four of those structures, the glycocalyx, flagella, fimbriae, and pylae, not all of them are found on all prokaryotes or on all bacterial cells. Different species will have kind of different combinations of those structures. Some of them might not have any of the structures at all. Some of them might have all of the structures, and others would have some combination. In this diagram here, this thick kind of blue-green stuff is actually a glycocalyx. Up here, the black stuff surrounding the cell is another glycocalyx, more of kind of a slimy one. This is more of a jelly one. All these little sticks pointing out of the cell are fimbriae. The longer stick is the pilus, and the longest kind of whip-like thing is the flagellum for this cell. Over here, you can see another bacterial cell that has the sticks coming out of it, so these are its fimbriae, and then it has several longer structures, which are its flagella, so this cell has multiple flagella. And over here, this cell in the middle is actually an E. coli cell. The stuff coming off of it are its fimbriae, and at the edges of the image, you can see 3 red blood cells. So the E. coli cell is actually using its fimbriae to attach to these red blood cells. The flagellum, again, would be used to move through liquid, and the pilus is used primarily to exchange genetic material, and the glycocalyx for protection. The last external structure to talk about is the cell wall, which would be between the cell membrane and the glycocalyx for a cell that has a glycocalyx. But it's always going to be, located outside the cell membrane. Almost all bacteria have a cell wall. There are a couple of exceptions, but almost all of them have a cell wall. The cell wall is for protection, of course. It also provides structural support, so it can help the cell maintain its shape in conjunction with the cytoskeleton. And it's also just going to protect the cell from toxins, but very importantly from osmotic stress. So if the bacterial cell ends up in an environment where it's actually hypotonic, so there's more solutes inside the cell and less solutes in the water outside of the cell, that would be hypotonic. That type of environment is actually really common to find. If the bacteria is in that type of environment, water is going to start entering the bacterial cell by osmosis. right? So water will always travel to wherever you have the most solutes, which in this case would be inside of the bacterial cell. So that cell is going to be filling up with water. And of course, it could actually theoretically swell so much that it would burst. But the cell wall is going to protect it from that, because the cell wall is really strong and it's really hard to stretch the cell wall. So the cell membrane can expand until it kind of hits up against the cell wall, and then it can't expand any more than that. So the cell wall is going to prevent the cell from popping if it gets into a hypotonic solution. That's kind of the main reason why bacteria have cell walls, is to protect them from popping in hypotonic solutions. In general, the cell wall for bacteria is made out of peptidoglycans, which we had gone over in the chapter on carbs. We talked about the structure of peptidoglycan, but again, that's a polysaccharide, just a long chain of glucose, sorry, not of glucose, of NAG and NAM sugars that are alternating. So it forms like a long straight chain, and then you take a lot of those chains and stack them next to each other, parallel to each other, and they will actually have interactions forming between the chains to hold them together, and that makes a very strong structure. There's two basic types of cell walls that bacteria can have that are made out of peptidoglycan. Almost every bacteria that has a cell wall has one of these two types. It could either be gram-positive or gram-negative. In gram-positive bacteria, the cell wall structure is really simple. It's just very thick layers of peptidoglycan. In gram-negative bacteria, it's a bit more complicated structure. It's only a thin layers of peptidoglycan that they have in their cell wall. Then on top of that, they actually have another membrane, which is called the outer membrane. And then the outer membrane has a particular molecule embedded in it, which is called lipopolysaccharide, or LPS. So these diagrams over here are showing the two types of cell walls. On the top, you have a gram-positive cell wall. So if you zoom in on it, all of this is the cell wall. It's basically entirely peptidoglycan, really thick layers of peptidoglycan stacked on top of each other. Then you also have some other proteins running through the layers to help hold them together. And then at the bottom is the regular plasma membrane of the cell. Then here you have a gram-negative cell wall. So the layers of peptidoglycan there are much thinner than they are in the gram-positive cell wall. And on top of it, you have another membrane called the outer membrane. These kind of squiggly things coming out of that outer membrane are actually the LPS molecules. So lipopolysaccharide has a little bit of it that is fatty. The lipo part, or maybe not fatty specifically, but that is a lipid. So that lipid part of the LPS is actually going to be embedded into the outer membrane, and then the polysaccharide part of it, which is a carbohydrate, is what you see sticking up here. So this LPS ends up being pretty important because the part of it that's a lipid that's actually embedded into the outer membrane is actually toxic. So that's kind of fine as long as the bacteria is kind of alive and intact, because the lipid part is embedded in this outer membrane, and so you're not actually exposed to it. But if the bacteria die, then their cell wall is going to break down, and the LPS will be released, and the lipid part of it will end up being exposed, and your immune system is just going to respond to it very strongly. So if you have an infection with gram-negative bacteria, and then you treat with antibiotics, if all those bacteria die at the same time, and you have all of their LPS released at the same time, that can actually be dangerous for you, because your immune system is going to have a really strong response to it. And if your immune system has just kind of too strong of an immune response, you can actually enter shock and die. And when that's happening, what's actually going on is that you just have inflammation all over your body. Inflammation is a major part of the immune response. And whenever you have inflammation, you end up having vasodilation, so your blood vessels get wider. And also the walls of the blood vessels become kind of easier for water in the blood to leave those blood vessels. And if that happens all over your body, basically it's going to drop your blood pressure. When your blood pressure gets too low to make your blood travel around your body, then you enter shock. like shock is when you don't have enough blood pressure to support blood flow to your organs, and that will kill you if it goes too far. So whenever someone is being treated for a bacterial infection, it's pretty important to know if they're infected with a gram-positive bacteria or a gram-negative bacteria. Part of the reason is just that gram-negative bacteria have the LPS, so you kind of want to be just a little bit more careful with how you use antibiotics with a gram-negative infection. But also, different antibiotics are going to be effective against different types of bacteria, and a lot of antibiotics are either good for gram-positive bacteria or for gram-negative bacteria, but not good for both. And that's because different molecules will be able to get through the different cell walls. So a molecule that gets through the gram-positive cell wall might not be able to make it through the gram-negative cell wall, and vice versa. The other reason is just that some antibiotics actually attack the cell wall directly, and of course the effectiveness of that attack depends on if the cell wall is the right type. So now we'll go through the structures that you find inside eukaryotic cells, including the membrane-bound organelles. So there's some internal structures in eukaryotic cells that are the same as internal structures that you find in prokaryotic cells, including the cytoplasm, cytosol, cytoskeleton, and ribosomes. So the cytoplasm for a prokaryotic cell is everything inside the cell. But for a eukaryote, it's everything inside the cell except for the nucleus. So that is a little bit different. Then the cytosol is just the same. It's the water in the cell, or the liquid in the cell that's actually more like a jelly, and everything that is dissolved in it. Then you have the cytoskeleton, which is basically doing the same job in prokaryotes and in eukaryotes, and in both types of organisms, it's a protein framework that is giving the cell shape and organization. But its structure in eukaryotes is a lot more elaborate than the structure of the cytoskeleton in prokaryotes. Also, there's an additional function of the cytoskeleton in eukaryotes, which is to help things kind of move along. So eukaryotes can use the cytoskeleton to transport substances within the cell from one part of the cell to another, and some eukaryotes can also use their cytoskeleton to move the entire cell. And then, of course, ribosomes do the same job in eukaryotes and in prokaryotes. But in eukaryotes, they do have like a slightly different structure. So you can tell if you have like the genetic code for a ribosome, you can tell if it's for a eukaryote or for a prokaryote. In prokaryotes, ribosomes just kind of float around in the cytosol. In eukaryotes, some of them are floating in the cytosol like that, but some of them are attached to another organelle. So you have some that are studying the nucleus, and some that are studying an organelle called the rough endoplasmic reticulum, which we will talk about in a moment. In this image of a eukaryotic cell, you can see the nucleus is in the middle. It's in yellow. And then in green around it, you have the cytoskeleton of the cell. So these protein fibers fill up the entire cell. They're so thick when you get close to the nucleus that you can't really tell them apart from each other. And then they kind of thin out as you go to the edges of the cell. But they're just filling up the entire cell. So the cell shape with these kind of little points over here, that's actually determined by the cytoskeleton. And then the cytoskeleton will also be used to move stuff around inside that cell. Now we'll go through the membrane-bound organelles that you find in eukaryotic cells. First, just a vesicle or a vacuole. A vesicle or a vacuole is just a sac of membrane that is storing some stuff. A vesicle is small, a vacuole is big. And that's kind of the main difference between them. So where in a prokaryotic cell you would have inclusions, in a eukaryote you're going to wrap that stuff up in a membrane and package it either as a vesicle if it's small or as a vacuole if it's large. Some vacuoles can be really, really large actually, especially in plant cells. In animal cells, if they have vacuoles, they're not that big. But the diagram over here is actually showing vesicles and vacuoles. Well, actually, maybe I should say just vacuoles. So this is a plant cell, and this really large structure inside of it is a vacuole. Every plant cell will have one really big vacuole in it, and then these are smaller vacuoles. They're actually still too big to be vesicles. Vesicles are really small, but they are, kind of small vacuoles. So in an animal cell, you can have vacuoles, but not that big. Then you have the nucleus, which is of course the defining organelle for eukaryotes. They're just defined as organisms that have a nucleus. Inside of the nucleus is where the DNA of the cell is stored. All of the DNA that's found in the cell is contained in the nucleus, and it's going to be arranged into different chromosomes. Depending on the eukaryote, that's going to determine like how many chromosomes are there, but it's probably more likely to be more than one. And it's all going to be packaged up. So it's going to be wrapped around histone proteins and then, additionally packaged on top of that. Some of it will be like super coiled. There's two kind of types of DNA in the nucleus that are kind of categorized based on how tightly they're packed. Anytime you have DNA that's packed with histone proteins, we call that chromatin. So we have euchromatin and heterochromatin inside the nucleus. So all of the DNA is packed around histone proteins. It's all going to be some type of chromatin. If it's euchromatin, then it's not really very tightly packed at all. So it would basically just be packed around the nucleus and then, packed around the histones and then maybe wound a little bit more than that, but basically just kind of around the histones. So very loosely packed. And then heterochromatin is going to be really tightly packed. So it's not just wrapped around the histones, but the histones are all kind of coiled up, and then you continue coiling it until you get supercoils in it. Apart from just how tightly the DNA is packed, euchromatin is also going to be like active regions of DNA, and heterochromatin will be inactive. And that's just because it's so tightly packed that the cell can't really send enzymes in there to access it. So if you're going to use some DNA and like have that DNA actually be active, you need to get enzymes up in there that can find the genes that you need to make protein with and actually build an mRNA copy of those genes. So if the DNA is super tightly packed, then there's no space to get the enzymes in there to build mRNA, and that DNA is effectively going to be inactive or dormant DNA. So euchromatin is loosely packed and active DNA, heterochromatin is tightly packed and inactive DNA. Most of the DNA is going to be heterochromatin in the nucleus, but the euchromatin is going to take up a lot more space because it's more loosely packed. Inside the nucleus, you also have a special region called the nucleolus. In the nucleolus, you're making your rRNA and you're assembling ribosomes. So the protein part of the ribosome is actually going to be made outside of the nucleus, and then you'd actually transport it into the nucleus in the nucleolus, and then stick it together with the rRNA to make actual ribosomes. The nucleus is an unusual organelle because it has two membranes around it. Most organelles will just have one. And we kind of talk about those two membranes around the nucleus as a single unit using the name nuclear envelope. So the nuclear envelope is just the two membranes that surround the nucleus. And periodically you have little holes in the nuclear envelope, and we call those nuclear pores. So anything that needs to get in or out of the nucleus is going to travel through one of those pores. Then you can also have ribosomes studying the nucleus. And let's see, this diagram here is kind of showing the structure of the nucleus. So you can see the double membrane surrounding it, which is the nuclear envelope. These little holes in the membrane are the nuclear pores, and the little pinkish dots are the ribosomes that are studying the nucleus. Inside the nucleus, you have this kind of egg yolk looking thing, which is the nucleolus. So your rRNA is being made there, your ribosomes are assembling there, then they would travel out of the nucleus through a nuclear pore. And the rest of the nucleus is filled up with chromatin, or DNA. So DNA wrapped around histones, we call it chromatin. The yellow parts are the euchromatin, which is loosely packed. It's active. The cell can use it to make proteins. In green, you have the heterochromatin, which is tightly packed and it's not active. So the heterochromatin is not taking up that much space because it's really, really tightly packed, but there's actually a lot of DNA in there, even though it doesn't take up very much volume. So typically we think about every eukaryotic cell as having a nucleus in it, but there actually are some exceptions to that. in your own body, you actually have red blood cells that don't have a nucleus at all. Most eukaryotic cells, of course, do have a nucleus, but there are a few exceptions, including your own red blood cells. And then there's also some cells that have more than one nucleus. An example of that would be your skeletal muscle cells. They're actually really big cells. Every skeletal muscle cell will usually run the whole length of the muscle. So if you have a muscle that reaches, all down your thigh, then that means that the cells in that muscle are basically as long as your thigh as well. So those are really big cells, and they actually need more than one nucleus to support the entire cell. Another really important membrane-bound organelle is the endoplasmic reticulum. Um, there's actually two kind of parts of the endoplasmic reticulum, and they're treated as being separate organelles. So you have the rough endoplasmic reticulum and the smooth endoplasmic reticulum. Both of them are going to be located around the nucleus, and it's kind of a complicated structure in this organelle where it's like multiple layers of membrane that are all kind of connected to each other in different ways, and also connected to the nuclear envelope. So in this diagram here, here you have the nucleus. The nuclear envelope is shown in green here. And this kind of tan stuff that's around the nucleus is the endoplasmic reticulum. So you can see the multiple layers of membrane here, and they're all connected to each other and also connected to the nuclear envelope. You can also see the two parts of the endoplasmic reticulum. The part that has all these little blue dots on it is the rough endoplasmic reticulum, and then the part that doesn't have those dots is the smooth endoplasmic reticulum. The rough endoplasmic reticulum is called that because it has ribosomes all over it studding its surface. So these little dots are actually ribosomes that are studding the surface of the rough endoplasmic reticulum, or the rough ER. And the rough ER is actually important for building proteins. So these ribosomes would actually be using mRNA to build amino acid chains, and then it's actually going to kind of release those amino acid chains into the rough endoplasmic reticulum. And you're going to have the proteins folding up within the rough endoplasmic reticulum. So all of your molecular chaperones that will help the proteins fold correctly are located inside of the rough endoplasmic reticulum. In addition to just folding the proteins, the rough ER is also going to modify them in different ways depending on what protein it is. And some of them are going to end up packaged into vesicles that will travel to the next organelle in the kind of the chain, which is the Golgi apparatus. And we'll talk about the Golgi apparatus right now. But first, we'll just briefly go over the smooth endoplasmic reticulum, which is just called that because it doesn't have ribosomes on its surface, so it doesn't look rough on its surface. The job of the smooth endoplasmic reticulum is to make lipids, and then it packages those lipids into vesicles and transports them around the cell, or sends them out to be transported around the cell. The smooth endoplasmic reticulum can also be used to store calcium. Calcium is a signaling molecule inside of most cells. And so some cells will keep kind of a repository of calcium in their smooth endoplasmic reticulum, or their smooth ER. One example of a cell that does that would be a muscle cell. So your muscles rely on calcium in order to contract. In order to contract, they have to release calcium. And that calcium is all stored in their smooth endoplasmic reticulum. So when they're going to contract, they release it. Then they pump it back into the smooth ER for the next contraction. But for a protein that was made by one of these ribosomes and ended up entering the rough endoplasmic reticulum and folding up in there, it's actually going to be packaged into a vesicle and sent out to the Golgi apparatus. So here you have proteins being put into a vesicle. This little ball thing, membrane ball, would be a vesicle, and that's going to go to the Golgi apparatus. The Golgi apparatus looks kind of similar to the endoplasmic reticulum because it's also layers of membrane, but the layers of membrane in the Golgi apparatus are not connected to each other. Each one is going to be separate from the others. Each one is going to have different enzymes inside of it. And this vesicle containing new proteins is going to get to the Golgi apparatus. It's going to kind of fuse with the membranes of the Golgi apparatus and release the proteins into the Golgi apparatus. Those proteins are going to be modified, and they're going to end up traveling through the Golgi apparatus from one layer to the next. They might not go through literally every single layer. It kind of depends on what the protein is. But at each stop, they're going to get modified again. Basically, what the Golgi apparatus is doing is giving these proteins little tags that are going to send them to their correct destination. So the Golgi apparatus is like a sorting station. It receives new proteins from the rough endoplasmic reticulum, and it sorts them and modifies them according to what destination they need to go to in the cell, and then it's going to package them into a vesicle and send them out to the correct destination. So that destination might be a specific organelle in the cell, or it might actually be the cell surface itself. And that's what's being shown here. So here you have a vesicle that is leaving the Golgi apparatus. It contains proteins that have been modified by the Golgi apparatus, and it's traveling to the surface of the cell where it's going to fuse with the cell membrane and then release the proteins outside of the cell. Not every single eukaryote has a Golgi apparatus, but most of them do. And then lastly, the other name for the Golgi apparatus is just the Golgi body. Another membrane-bound organelle found in a lot of eukaryotes is the lysosome. It's actually only found in animal cells. The job of the lysosome is just to break stuff down and kind of recycle things. So it's just a little sac enclosed by membrane. It's very acidic in the interior, and it has a lot of enzymes that break stuff down. So whenever you have a component of the cell that is kind of damaged or worn out, it's actually going to be taken to a lysosome to be degraded, broken down into its component parts, and then those components will be recycled by the cell. Also, sometimes if the cell takes something in from its environment, maybe a bacterial cell. There are some animal cells that will actually eat bacterial cells. So if that happens, it's actually going to send the bacterial cell to a lysosome to be destroyed, and then it will use the components of that cell. You also have peroxisomes in some eukaryotes. A peroxisome is a little bit similar to a lysosome, at least in how it looks. It's really similar, so it's also just a small sac of membrane, but the main difference is that it has different enzymes from the lysosome. So the peroxisome is going to have enzymes that do redox reactions, and the basic point of it is going to be just to react with any reactive oxygen species that are around or other types of toxins to make them less toxic. So a reactive oxygen species is abbreviated ROS, and it's basically just a molecule that has oxygen in it and is very reactive. Usually a reactive oxygen species will have an unpaired electron in it somewhere. Anytime you have an unpaired electron on an oxygen, that is super reactive, because oxygen is really electronegative, so it's already going to grab electrons really easily and pull them in towards itself, but also an unpaired electron is not very stable. If it can gain another electron, so it'll be part of a pair, then it'll be much more stable. So a reactive oxygen species is just really good at stripping an electron off of some other molecule. Which would mean that it's really good at oxidizing other molecules and stealing their electrons. So you actually will produce reactive oxygen species normally in your metabolism. So in order for your body to create ATP, it also has to create a small amount of reactive oxygen species as a byproduct. Since you're always producing small amounts of these reactive oxygen species, you need to have a way to kind of deal with them and detoxify them. If you don't, they're going to end up reacting with any molecule that they encounter in the cell. If they react with a protein, there's a good chance that protein won't work right afterwards. If they react with your DNA, then they can cause mutations and even cancer. So it's really important for your cells to be able to detoxify those things, and they will do it using the peroxisome. So you send reactive oxygen species into the peroxisome, and they're going to react with some other molecule that is going to give them an electron and reduce them. Once they've picked up an electron and have been reduced, they're not going to be reactive anymore, so they're going to be like a lot safer. In this image, you can see both lysosomes and peroxisomes. The lysosomes are darker kind of little sacs. Here's another one. And then the peroxisomes are lighter little sacs. But otherwise, you can see they're pretty similar in their structure. Just a small sac enclosed by enzymes. Enclosed by a membrane that contains enzymes. The last two organelles that we'll talk about in eukaryotes are mitochondria and chloroplasts. Mitochondria have two membranes around them, so they're unusual because, like the nucleus, they have two membranes, and their function is to produce ATP. Mitochondria is actually the plural word, and the singular is mitochondrion. Chloroplasts are kind of similar. They also have multiple membranes. They actually have 3 membranes, not just 2, and they also produce ATP. But they don't just produce ATP, they also produce sugar and are the sites of photosynthesis. So later on in the semester, when we talk about metabolism, we're going to come back to mitochondria and chloroplasts. Mitochondria are where you're doing a process called cellular respiration, where you're using glucose to make ATP. In chloroplasts, you're doing photosynthesis and you're using light to make ATP and sugar. A mitochondrion is shown in this diagram here. So they're kind of cylinders. Some of them are a little wiggly, some of them are shorter and squatter, so they can have kind of different shapes, but they're all basically cylindrical. They have one membrane that just goes around the entire organelle, and then the inner membrane has little folds that enter the lumen of the mitochondrion. Those folds are called cristae, and then we also have a special name for the lumen of the mitochondrion, which is the mitochondrial matrix. So in between these cristae is where you have the mitochondrial matrix. It's actually in that matrix and in the membranes of the cristae where you are making ATP. And we'll go into the details of that process later on in the semester. Then for chloroplasts, that's shown on the top. You have actually 2 membranes that enclose the chloroplast on the outside, just running around the outside of it. And then you have the third membrane, which is inside the chloroplast. And that third membrane has a really complicated structure. So it's actually formed into these discs. Each disc is called a thylakoid, and then those discs are also stacked up on top of each other. Every stack is a granum. So in these grana is the plural of granum, that's where you're actually capturing light and making ATP in photosynthesis. So all of these grana are connected to each other by this inner membrane inside the chloroplast. Then you also have the kind of space between, or the space outside of the grana that is enclosed by these 2 outer membranes, but it's not enclosed by the inner membrane. That space, which would be like the lumen of the chloroplast, also has its own special name, and it's called the stroma. In the stroma is where you're actually making the sugar from photosynthesis. Most eukaryotes have mitochondria, but only plants and algae will have chloroplasts. So most eukaryotes do not have chloroplasts. And even the eukaryotes that do have chloroplasts, the plants and the algae, they're actually getting most of their ATP from their mitochondria. So they're using their chloroplasts to do photosynthesis, and what they're mostly getting out of that is sugar. Most of the ATP that they make in photosynthesis, they have to use it to actually make the sugar. And then the sugar gets used by the mitochondria to make ATP and to build other things in the cell. It gets used by structures other than the mitochondria to build things in the cell. Mitochondria and chloroplasts are kind of really special organelles. They're not like any other organelles. Of course, they have multiple membranes around them, which is very unusual, but they also have other features that are unique. So they actually contain their own ribosomes and their own DNA, and they don't divide with the nucleus. So usually when the cell is going to divide, it kind of prepares for that by copying all its DNA and making double of its organelles, or just kind of, doubling the size of its organelles. And then at the very end, the nucleus splits into two, and the whole cell also splits into two. So all the organelles are getting split along with the nucleus. But with the mitochondria and the chloroplasts, they actually are dividing kind of on their own schedule just throughout the life of the cell. Then whenever the cell is going to divide, one of the new cells kind of ends up with some of the mitochondria and chloroplasts just by random chance, and then the other daughter cell will get the rest of them. That DNA that you find in the chloroplast and in the mitochondria is actually going to be inherited through the through the maternal line only. In eukaryotes. So you have a sperm cell and an egg cell basically. I mean, in other species it's a little bit different, but you're always going to have some analog of the egg cell that is actually containing like all the parts of a cell that has all the organelles and stuff. Then a sperm is just a really stripped down cell. There's not much to it at all. It mostly just has the nucleus and then its tail to let it move. So it doesn't have like mitochondria or any way of like producing its own ATP and stuff. Not really. So actually the mitochondria for a fertilized egg is all coming from the original like mother's egg. So all of the DNA that those mitochondria have is, the mother's mitochondrial DNA. And then all of the other cells in your body are coming from that original cell. So all of the mitochondria that you have, the DNA that they have is actually inherited from your mother and not from your father. Only the DNA in your nucleus is actually coming equally from both your mother and your father. So because mitochondria and chloroplasts are such unique organelles, we actually kind of think that they didn't start off as organelles at all. Endosymbiotic theory is basically the idea that mitochondria and chloroplasts used to be independently living bacterial cells that got engulfed by an early eukaryotic cell, and instead of just dying there, they actually kind of survived and prospered and kept dividing inside of this eukaryote and ended up just kind of evolving into organelles. So some of the evidence to support endosymbiotic theory is the fact that mitochondria and chloroplasts have their own DNA and their own ribosomes and divide independently of the rest of the cell. So it's weird for an organelle to have DNA. All the DNA in the cell is stored in the nucleus. If mitochondria and chloroplasts have DNA and have ribosomes, they have all the basic machinery they need to make proteins. It's like they used to be it's like they used to be independent organisms that made their own proteins. And when you look at their DNA, it actually doesn't look like eukaryotic DNA. It looks like bacterial DNA. And when you analyze the sequences of that DNA, you can actually tell which bacteria mitochondria and chloroplasts are specifically related to. So mitochondria are actually related to proteobacteria, and chloroplasts are related to cyanobacteria, which are just two different types of bacteria. But the fact that you can actually see those sequence similarities between mitochondria and chloroplast DNA and those specific lineages of bacteria, it's really, really strong evidence to suggest that those used to be independent organisms, and they got eaten by an early eukaryotic cell, and ended up becoming organelles. And the process might have gone something like this diagram. So we don't really know exactly how eukaryotes emerged, but in this diagram we have an early eukaryote that already has a nucleus and some basic organelles. And it is going to end up engulfing a proteobacterium. Normally, when a eukaryotic cell engulfs a bacterium, it's going to send it to a lysosome and destroy it. But in this case, this proteobacterium does not get destroyed. Instead, it just starts floating around in this cell, and it will divide and end up just, basically living inside the cell. Eventually, it converts to a mitochondrion. So it's basically losing the ability to live independently, and it's becoming dependent on the host cell that's around it, to the point that it's not an independent organism anymore, it's just an organelle. And of course, the host cell is benefiting by getting all the ATP that this bacteria is producing. Then at some point later in the evolution of eukaryotes, you would have had another eukaryotic cell engulf a cyanobacterium, which is a type of bacteria that's able to do photosynthesis. So again, instead of the cyanobacterium being sent to a lysosome to be destroyed, it actually accidentally ends up kind of floating around inside the cell and not dying. And it's getting a nice stable environment inside of this cell. It's a much nicer place to live than, the outside. So it ends up prospering there and dividing and basically losing its independence and becoming a chloroplast. And again, the eukaryotic cell is benefiting by getting the photosynthesis. It gets all the sugar that this cell is producing, although this cell is now an organelle. So this idea, endosymbiotic theory, is not universally accepted by every single scientist, but it is very widely accepted and very well supported, which is why it's called a theory and not a hypothesis. So we'll talk real briefly about some of the external structures of eukaryotic cells. Some of them actually do have glycocalyces like what bacteria can have, but we won't really worry about that. We'll just focus on their cell walls. So a lot of eukaryotes have cell walls, kind of for the same reason that bacteria have cell walls. So they're designed to give protection to the cell and to help it, maintain its shape. And again, the main type of protection that this cell wall is providing is protection against hypotonic environments, because cells are usually in a hypotonic environment where there's more solutes inside the cell and less solutes in the water around them. And then water would enter the cell by osmosis and the cell would swell up and pop. But for most eukaryotes, they will not actually pop in a hypotonic environment because of their cell wall. They can't swell to be larger than what their cell wall is because it's very strong and it doesn't really change its shape. So for plants, their cell walls are made out of cellulose, as we kind of mentioned in the carb chapter. For algae are single-celled eukaryotes that can do photosynthesis. They actually have different things that can form their cell walls. Some of them have cell walls that are made out of agar, some have carrageenan, or I should say carrageenan. Some actually have silica in their cell walls. And this image here is actually algae that have silica in their cell walls. Silica is kind of like glass. So you can see they have a lot of really unique structures in here that are formed by the cell wall, and a lot of pretty colors. It's just very pretty. The ones that have the silica cell walls are really cool. Then you have fungi, which, as we mentioned, have cell walls made out of chitin. Animal cells do not have cell walls. Instead, they will produce extracellular matrix, which is basically just proteins and other, also some carbohydrates that they produce, and then they stick it outside of the cell, and it ends up forming kind of this like scaffolding that the cell is immersed in. The purpose of that extracellular matrix is to kind of help these cells be organized into a tissue and help them stick together. So cell walls can also help cells stick together, but for animal cells they don't have that, so they need an extracellular matrix instead. But of course, the extracellular matrix is not going to protect them from a hypotonic environment. It doesn't prevent them from swelling up at all. It just kind of connects them to each other. So in order to protect themselves from swelling up and popping in a hypotonic environment, animal cells will use the sodium-potassium pump. The last thing we'll kind of talk about in talking about the structure of eukaryotic cells is just a basic comparison of animal and plant cells. So for plant cells, they have a cell wall and it's made out of cellulose. But for animal cells, they don't have a cell wall at all. That means that plant cells end up having a rigid shape. They can't change the shape. The shape is just determined by the cell wall. But for animal cells, they actually can change their shape. And there's a lot of different shapes that they can form. Their shape would actually be determined more by their cytoskeleton compared to their cell wall, because they don't have a cell wall, of course. Then in plant cells, you have both chloroplasts and mitochondria. In animal cells, you only have mitochondria. In plant cells, you always have a large central vacuole. You can store stuff in that vacuole, but also the job of it is to kind of store water inside the cell so that the cell can make sure it always has sufficient water. In animal cells, you don't have a really big vacuole like that. You might have some smaller vacuoles, but not a really big one. And then just in plant cells, you don't have lysosomes, but in animal cells, you do have lysosomes. Actually, for some plant cells, they're going to be using a vacuole instead of a lysosome. So they might have a vacuole that is acidic like a lysosome and is basically breaking things down kind of the way that a lysosome would do, but it's not. It's not a lysosome. So here you have kind of a visual comparison. In yellow on the plant cell, you can see the cell wall wrapped around it that is determining the shape of that cell. And here you have the animal cell. It doesn't have a cell wall, so it has a much more irregular shape, and its shape can actually change. These kind of whitish fibers inside of the animal cell are its cytoskeleton that's actually giving it its shape. These kind of yellowish fibers outside of it, that's the extracellular matrix. So that's just going to be attaching that animal cell to other cells that are around. For the plant cell, you can see it's actually connected to surrounding cells by the cell wall itself. You can also see inside of the plant cell both the chloroplast and the mitochondrion, or mitochondria. In the animal cell, you see mitochondria, but you don't see any chloroplasts. In the plant cell, you can see the large central vacuole, where it's going to be storing stuff, but most importantly, water, storing water. In the animal cell, you do not have a large central vacuole. You might have some smaller ones around somewhere. Then finally, in the animal cell, you have lysosomes, and in the plant cell, you don't. So this little ball here is a lysosome. In the plant cells, you don't have any. You do have this ball, but that's a peroxisome. So you have those peroxisomes in both plants and animal cells, but lysosomes are only in animal cells. Okay, so now we've gone over the structures that you find in a eukaryotic cell. We're going to finish the chapter by talking a little bit about how some of those structures work together to support the functions of the cell. So first we'll talk about nuclear traffic. That means moving things in and out of the nucleus of the cell. The nucleus is surrounded by actually by two membranes that together are called the nuclear envelope. And in the nuclear envelope, you have these pores called nuclear pores, and each of those pores has a complex called the nuclear pore complex. It's made of a bunch of proteins, and its job is just to regulate how things are moving in and out of the nucleus. So whenever things need to go out or in, they have to go through that pore and interact with the proteins on that make up the pore. So some things that need to get moved out of the nucleus or exported from the nucleus would be, of course, mRNA, and then also tRNA. So all of your RNA is actually made inside of the nucleus, and all of it, well, most of it needs to leave the nucleus in order to function or to have its function. So that includes your mRNA and your tRNA. As you remember, tRNA we mentioned in lab. It's involved in translating mRNA into protein. So the ribosome will use tRNA to bring in the correct amino acid that needs to be added next to the protein. rRNA also, that needs to come out from the nucleus. It's made inside of the nucleus. Actually, ribosomes themselves are assembled inside the nucleus in part of the nucleus called the nucleolus. So you actually would make your proteins that are going to be part of the ribosome outside of the nucleus, where you make all of your proteins. Then they have to get sent back into the nucleus and assembled into a full ribosome with the rRNA in the nucleolus. Then that ribosome needs to be exported again out of the nucleus. So that's most of the stuff that needs to come out of the nucleus. You also have stuff that needs to go in or be imported. That would be, of course, nucleotides, so that you can make your DNA and RNA. Also, any proteins that are needed in the nucleus. So all the proteins that attach to DNA so it can be packaged, all the proteins that are going to go into the ribosome. When we talk about how genes work later in the semester, we'll mention polymerase enzymes. Those are enzymes that make DNA or RNA, so those need to go into the nucleus. Also, proteins for the nuclear pore complex itself need to be transported into the nucleus. All of that is strictly controlled. If you have like a small molecule like an ion or water, that can go through the pore just by itself, no problem. But any large molecule that's going to move through the pore has to interact with the nuclear transport proteins in order to get in or out. That process requires ATP. So the diagram there is showing how things are imported or exported, how they're attached to the different nuclear transport proteins. You really don't need to know the details of any of that. It's just for your interest. Any protein that is going to be entering the nucleus has to have a special tag that's going to send it to the nucleus. That's called the NLS, or the nuclear localization signal. That's a tag that's made out of amino acids. So once the protein is made, it would enter the rough endoplasmic reticulum and be modified. One of the modifications would be adding that nuclear localization signal onto it so that then it will be transported into the nucleus. Hold on, I'm getting a call. Alrighty. Oh, where was I? Oh boy. Okay, so for any protein to get into the nucleus, it needs to have the nuclear localization signal attached to it. So make sure you're aware of that. Then you also have a different signal that's used to export things from the nucleus. So anything that's any protein that's going to be leaving the nucleus has to have the nuclear export signal, or the NES, attached to it. That's another amino acid tag. So in research, when you want to make sure that a protein that you're studying enters the nucleus of the cell, you can attach the nuclear localization signal to it. That will direct anything that gets it into the nucleus. Just FYI. Then you also have the endomembrane system, which is a system that includes several different organelles as well as the cell membrane itself. Its job is to move proteins around in the cell and sort them so that you make sure they go to the right spot. It includes the rough endoplasmic reticulum, the Golgi apparatus, lysosomes, and the cell, the whole cell membrane. All of those things are included in the endomembrane system because they kind of share membranes with each other. Whenever you have some protein that's going to be moving around in that system, let's say it starts off in the rough endoplasmic reticulum, which is where it would generally start, it's going to leave in a vesicle. That vesicle is actually made from the membrane of the organelle that the protein is coming out of. So in this case, it would be made of the rough endoplasmic reticulum membrane. Then that vesicle is going to travel to the next organelle in the system, which is the Golgi apparatus. It's going to fuse with the membrane of the Golgi apparatus. So now this little chunk of membrane that is from the rough endoplasmic reticulum is part of the membrane of the Golgi apparatus. The proteins will be released into the Golgi apparatus. They'll travel through the Golgi apparatus and end up leaving in another vesicle that is made of the membrane of the Golgi apparatus. Then it can go to different places. It could go to a lysosome. It could also go to the cell surface. For proteins that are going to be excreted out of the cell, those would be sent to the cell surface. The vesicle is going to fuse with the cell membrane by exocytosis, and the contents are released. So that means that the membranes of all these parts of the endomembrane system are connected to each other. They're all sharing their membranes with each other, and that's why they're all counted together in the endomembrane system. At every step along that path, the protein is going to be modified so that it'll be sent to the next location. In order to enter this endomembrane system, a protein needs to have the ER signal sequence, which is an amino acid tag, again, that is added to the protein as it's being made. Actually, it would be the first 20 amino acids of the protein would be the ER signal sequence. That could be abbreviated the ERSS, actually. So here you have a ribosome. This is the mRNA. It's reading the mRNA to build the protein. So you would have tRNAs coming into this ribosome and dropping off their amino acids. Those amino acids are getting added to this growing chain of the new protein. The first amino acids are this ER signal sequence, and then you have the rest of the protein being made behind it. in order to get into the rough endoplasmic reticulum, the first organelle of the endomembrane system, you have to have this signal recognition particle, or SRP, that's another protein, come and grab the ER signal sequence. Then this SRP protein is itself going to grab a receptor for it that is located in the membrane of the rough endoplasmic reticulum. So now you have this newly forming protein actually physically attached to the rough endoplasmic reticulum by the SRP and the SRP receptor. The SRP receptor is always found next to this pore called the translocon pore. Actually, if I go to the next slide, I think I have words there. There you go. So, all right. So your SRP receptor is attached to the translocon pore, and the protein, the newly forming protein, as it's still being built by the ribosome, is going to end up being pushed through this pore into the rough endoplasmic reticulum, so that as it grows, you have a bigger and bigger loop of the protein inside of the rough endoplasmic reticulum. When the protein is done, everything's going to detach, and the protein will be left inside the rough endoplasmic reticulum, and it's going to fold up. You'll have molecular chaperones that attach to it to help it fold correctly, and now you have your finished protein inside the rough endoplasmic reticulum, and therefore inside the endomembrane system. In the rough endoplasmic reticulum, after this protein folds, it's going to be modified. Generally, oligosaccharides, or little sugars, would be added to the protein. That makes it a glycoprotein. Any protein that has sugar attached to it is a glycoprotein. The glyc means sugar, and the protein is the base word, so that tells you it's a protein with a little bit of sugar attached to it. Those sugars that are added are telling the protein where it needs to go. You also have some proteins that aren't going anywhere else. They're actually going to stay in the rough endoplasmic reticulum. For instance, the proteins that are making up that translocon channel and the SRP receptor, those need to stay in the rough endoplasmic reticulum. So they're going to stay there. The ones that are moving on are going to be modified, making a glycoprotein. They're going to have a special oligosaccharide added to them that is a signal that sends them to the Golgi apparatus. So once they have that oligosaccharide added that sends them to the Golgi apparatus, they're going to be put inside of a vesicle, and that vesicle moves to the Golgi apparatus and fuses with the membrane of the Golgi apparatus. The Golgi apparatus has a bunch of layers of membrane that actually aren't really attached to each other. Each of these layers is called a cistern. So altogether, plural, those are cisternae in the Golgi apparatus. Each one of these layers, each cistern, has its own kind of chemical environment with its own enzymes inside of it. So each layer is chemically different and has different enzymes from the other layers. As the proteins are entering the Golgi apparatus, they're exposed to the enzymes in that first layer, or that first cistern, and they're going to be modified in different ways depending on which part of the cistern they are in. And they're going to be sent on in a vesicle to the next little layer. So that's, of course, a real short trip. And they're just going to be moving through the layers of the Golgi apparatus. Each time they move into a new layer, they're going to be modified in different ways. And different proteins are going to be going to different places ultimately. So they can move between different layers. So not every protein goes through every single layer of the Golgi apparatus. Some of them skip some layers. And they also will leave the Golgi apparatus at different points. So here you can see some proteins leaving here to go to a lysosome, some proteins leaving here to be stored in a vacuole, some proteins leaving on the other side to go to the cell surface. So a lot of the time, one end of the layer will actually be different from the other end of the layer also. And I didn't mention it in the PowerPoint, I guess, but you also have some proteins that need to stay in the Golgi apparatus. They're just going to go to whichever layer they need to be in, and they're going to stay there. At the other side of the Golgi apparatus, your proteins that are moving on will leave in another vesicle and go to their next destination. They are sent to their next destination, to the right next destination, because of those modifications that they've received. And it can be all different types of modifications. I think I've got that on the Slide. Yes. Cool. Um, Right, so most of the time it's carbohydrates, so different oligosaccharides that are going to be added to those proteins, or the ones that they already have on them from the rough endoplasmic reticulum can be modified by the enzymes in the Golgi apparatus in different ways. So you're going to get a lot of different oligosaccharides out of that. Every oligosaccharide is telling the protein where to go, or telling the Golgi apparatus, that is, where to send the protein in the vesicle that it will leave in. So each one of those oligosaccharides is a signal that is used to send the protein to its correct destination. And just as an example, if you were going to send a protein into the lysosome, maybe it would be a digestive enzyme protein that needs to go into the lysosome. You would need to add an oligosaccharide to it that has a mannose sugar within it, and that mannose sugar has to be modified so that it has a phosphate group attached to carbon 6. That's the signal that will send it to the lysosome. Any protein that gets that signal that has an oligosaccharide with a mannose with a phosphate group attached to the 6th sugar, that's going to go to the lysosome. If it needs to go to a different place, you would have a different oligosaccharide with maybe different modifications on it. So the figure there is showing some of the different oligosaccharides that are added to proteins in the Golgi apparatus, as well as different parts of the Golgi apparatus where they're added. So for these guys over here, to get these types of signals, you need to go through this side of the Golgi apparatus. For these guys right here, to get the signal, you need to go through the middle of the Golgi apparatus, and so on and so forth. You don't need to know those details, just that those different oligosaccharides and differently modified oligosaccharides are the signals that send the protein to its final destination. And finally, exocytosis and endocytosis. So when things are going to be leaving the cell, when large things are going to be leaving the cell, they need to go out through a vesicle by exocytosis. So if you have a small molecule like water or an ion or something like that, you're going to have a channel in the membrane or a transporter protein in the membrane to take it across. But if you have like a whole protein, proteins are real big. They can't go through a little channel or another protein. They need something more large. So they will leave in a vesicle. So here you have a vesicle full of proteins that need to be excreted onto the surface of the cell. This vesicle has come probably from the Golgi apparatus, and it's going to move to the cell surface and fuse with the membrane of the cell. Once it's fused, you get a little pocket like this. All these proteins that used to be inside of the vesicle are now outside of the cell, and then this membrane is just going to smooth out so that this vesicle is now part of the overall cell membrane, and the proteins that were inside of it are outside. That's exocytosis. Anytime the cell needs to secrete something large, it does it by exocytosis in general. You also have the opposite of exocytosis, endocytosis, for bringing big things in. So if you have something large that needs to be taken into the cell that can't fit in a channel or a carrier protein, you're just going to kind of have that thing cozy up to the surface of the cell. The cell membrane is going to make a little pocket for it, and then the pocket is going to pinch closed on the top. And when that happens, you end up with a vesicle that has that little thingy inside of it. That's endocytosis. It's basically the opposite of exocytosis. There's also a special type of endocytosis called phagocytosis. Phagocytosis is basically the same process as endocytosis, but it's for things that are like real big, like a whole cell, like a whole bacterial cell. So cells in your immune system that protect you from bacteria, some of them do that by eating the bacteria and then destroying them. Those are called phagocytes, and they bring in the bacteria by phagocytosis, by making a little vesicle that the bacteria ends up in and ingesting that vesicle. Finally, we'll talk about the lysosome. So you don't need to remember all these steps, but all these steps are not on the test, but we'll go over them for interest. So the lysosome is the main way that the cell gets rid of old stuff. is the main way that the cell digests things and recycles them. So anything that's in the cell, an old protein, an old organelle, that's going to be gotten rid of using the lysosome. Also cells that are kind of cleaners, like actually a lot of those immune cells that protect you from bacteria, they don't only eat bacteria, but they will also eat like pieces of your own dead cells that kind of got worn out, just cleaning up debris. So all those things are going to be digested inside of a lysosome. So if we're talking about something that has come from outside of the cell, little bits that need to be digested in the lysosome, those are going to be taken in by endocytosis or by phagocytosis if we're talking about bacteria. That ends up in a vesicle that's called the endosome. Actually, at first we call it the early endosome. In the membrane of this vesicle, you have a lot of proton pumps, and inside of it you have whatever stuff you're going to be degrading. All those proton pumps are taking hydrogen ions, which are protons, from the cytoplasm of the cell and pumping them, or from the cytosol of the cell and pumping them inside of the early endosome. As that process continues, the endosome becomes more and more acidic until it forms the late endosome, which has a very low acidic pH. Then you have your digestive enzymes that were made. They would actually be made, by a ribosome, then end up in the rough endoplasmic reticulum in the endomembrane system. They would be modified, so they're sent to the Golgi apparatus. They move through the Golgi apparatus, getting modified at each stop. Then they leave the other side of the Golgi apparatus in another vesicle that is going to get fused with this vesicle, with the endosome, with the late endosome. And that forms the final lysosome. So you have your digestive enzymes added to the low pH environment that they need to function appropriately, and then anything that was inside of there is going to get chewed up. And I think that's it. No, it's not it. Haha. Okay, cytoskeleton. The cytoskeleton we had mentioned is the framework that gives the cell shape and organization, but it's also important for transport in the cell, especially for vesicles. Anytime vesicles are moving around in the cell, they're not just floating through the cell, they're actually attached to the cytoskeleton and they're being dragged along. So here, this is an electron microscope image, and you can actually see these fibers, this one and also this one. These are fibers of the cytoskeleton. Those are part of the cytoskeleton. And then you have little bumps here. Those would be actually vesicles getting dragged along the fibers of the cytoskeleton to their destination. And then this diagram shows how they're walking. So you would actually have this microtubule here, that's a fiber, that's part of the cytoskeleton. Here's the vesicle that has proteins in it that are being transported inside the cell. And here you have this kinesin protein that actually grabs the vesicle on one end, and on the other end it uses ATP to walk along the microtubule fiber. And that's how vesicles are moved around inside the cell. So you don't really need to know the details on this too much, just that the cytoskeleton is used to transport vesicles inside the cell. But you do have different types of protein fibers in the cytoskeleton. Some of them are made of actin, some of them are made of tubulin, some of them made of keratin. The microtubule in that diagram is made out of tubulin. You have kinesin that is able to walk along the tubulin fibers, and you also have another protein, myosin, that's able to walk along the actin fibers. So kinesin is usually used to move things inside the cell, but then myosin would be used to actually move the cytoskeleton itself and kind of change the shape of the cytoskeleton. When that happens, the shape of the whole cell is going to change. So if you've kind of heard of amoeba that move by like sticking out a little foot from their membrane and attaching it to a surface and then pulling themselves up towards it, that's all based on myosin moving along actin filaments. Myosin moving along actin is also the way that your muscle cells are able to contract so that you can move.